Lipid absorption is a bit of a chemistry problem. So we have to look at the transporter activity and how these transporters actually work in order to transport and effectively absorb lipids from the lumen. So we have our triglycerides digested into our 2 fatty acids plus 1,2-monoglyceride by lipase activity. And these fatty acids and the fatty acid attached to the 2-monoglyceride structure, dietary fatty acids, tend to be C16 to C22. And this means that there's 16 to 22 carbons long generally. These are considered long to very long chains of fatty acid. Now, being a long chain fatty acid, this is where we have our chemistry problem. It would be easy to speculate that something of this long hydrocarbon chain would be very, very hydrophobic and would pass through the membrane and into the cell easily with no assistance required. However, remember the fatty acid structure has a carboxylic acid on the end of it. And this carboxylic acid is charged, deprotonated at neutral pH, and so this prevents the fatty acid from generally crossing the membrane. Now, the exception to this are short-chain fatty acids. Short-chain fatty acids, we're talking C5 to approximately C12, 5 to 12 carbons long on this fatty acid chain. These can pass the membrane. So they can pass the plasma membrane and get into the cell because they have just the right balance, kind of like Goldilocks and the Three Bears. They have just the right amount of hydrophobicity, just the right amount of hydrophilicity, that they need no transport. And so they can pass through by simple diffusion. Now, the long and very long chains, if these enter the membrane, they're going to get stuck because of the hydrophobic binding properties of these long chains hydrocarbons. So what we need is a transporter. And these transporters, there's many of them, but they're generally called FATP for fatty acid transport protein. These transporters work by binding the fatty acid from the outside of the cell and transporting it to the inside of the cell by facilitated diffusion. So there's no active transport involved in this process. This is simply working by a gradient of fatty acids being higher concentration in the lumen of our intestine and passing into the cytosol of the enterocyte in the intestine. Now, because this is facilitated diffusion, the reverse reaction to allow fatty acids to exit the cell is equally possible. So how do we prevent that? Well, we have another enzyme that is associated with the FATP, FATP, and this enzyme is called fatty acyl CoA synthetase. The purpose of this enzyme is to take the fatty acid that is transported in by the FATP and add a coenzyme A to the carboxylic end. So coenzyme A, many of you will recall this, call it CoAsh. This will be attached to the end. What this effectively does is it prepares the fatty acid for downstream activities that the cell needs to process, and it also allows for what's called trapping. Trapping means that the fatty acyl-CoA structure is no longer able to pass backwards through the FATP transporter, because the FATP transporter only recognizes fatty acids, not fatty acyl-CoA. Now, once again, we have a chemistry problem, and what we have to do is we have to protect the cell from this hydrophobic group on the fatty acyl. So what we have is, next step is FABP, which stands for fatty acyl binding protein. This can be considered a chaperone. The chaperone properties is it's going to bind the fatty acyl-CoA, and it does so like a clamshell. So if I draw a little Pac-Man structure here, the fatty acyl-CoA group fits nicely into the cleft of the FABP, and then this whole unit here, once it is bound, is soluble in the cytoplasm and can freely diffuse to the cytoplasm. Now, once we have our fatty acyl-CoA, what do we do with this fat to get it into the blood and transport it so that the rest of the tissues of the body has access? Well, again, we have to account for a chemistry problem. So with these fatty acyl-CoAs now inside the cytosol, they're going to be transported by FABP to the smooth endoplasmic reticulum. And in the smooth endoplasmic reticulum, we are going to recombine the fatty acids. So we are going to take 2 fatty acyl-CoA groups plus 1, 2 monoglyceride structure. And what are we going to make? We are going to make a triglyceride, and releasing our 2 coenzyme groups. Now that we have this triglyceride restructured, we are ready to package up this very hydrophobic molecule into a structure that can be easily and safely transported in the body. Again, also in the smooth endoplasmic reticulum, the triglyceride will be combined with a protein being made by the enterocytes of the intestine. That protein is called ApoB48. Apo stands for apolipoprotein. The ApoB48 protein serves as a skeleton, as a scaffold, and it has lots of hydrophilic and hydrophobic domains to it so that it can associate with the triglyceride and then form a larger lipoprotein structure. To this, we are going to add our cholesterol. We are also going to add our cholesterol esters. Recall that these were generated from our digestion. We're going to recombine the cholesterol with a fatty acid to make an ester, and any phospholipids that were obtained in our diet will also be added to this. The final product is called a chylomicron. Chylomicrons are very, very, very large structures. They have a hydrophobic core, the ApoB48 protein is located within this hydrophobic core, Cholesterol tends to be on the outside. Cholesterol esters are inside because these are fully hydrophobic. Anything that has polarity to it, such as a cholesterol with that 3-hydroxy group, or the head group of a phospholipid, will be associated with the outside. Now these structures are secreted out the bottom of the cell, the basal surface, and they are too large to enter the bloodstream. So these enter the lymphatics. Recall the lymphatics is the secondary vascular system, and these endothelial cells are leaky, which means that they have large junctions between them, allowing larger components to enter this vasculature. The lymphatics then enter the normal systemic circulation through the vena cava.